Good day, microbiology fans. I'm a realistic fish head, and today we are going to be talking about applied microbiology. Let's turn your attention to the screen in the center of Bikini Bottom. Our learning objectives for today is to explain how microbes function in biogeochemical cycles, explain the use of microbiomes in bioremediation, explain microbes' use in water purification and food production. We're going to identify ways to control harmful microorganisms in food and explain the importance of agricultural microbiology. The first thing that we're going to talk about are biogeochemical cycles. Biogeochemical cycles are simply pathways by which chemical substances move through the biotic and abiotic compartments of the earth. There are 5 primary biogeochemical cycles: carbon, nitrogen, sulfur, phosphorus, and water. There is a discussion page on Canvas for you to be able to identify where microbes play a role in this. I have images set up with the blocks placed in. You need to identify the location of those microbes and their purpose, and then post your answers to the discussion. Bioremediation is using microbes to transform harmful substances into non-toxic compounds. This can be bacteria that eat oil, or any toxic sludge. There are actually a number of very cool microbes that are being used right now in bioremediation. I would like you to learn these by name. You need to know the scientific names of the next 3 bacteria. The first is Alkanovix, which is an oil-eating bacteria. So when an oil spill occurs, these bacteria, which are always present in the environment, have a massive population explosion. This massive explosion allows them to feed on the oil because now they have their primary food sources in gobs and gobs and gobs. They will surround an oil droplet and start releasing different enzymes to help them break that oil down so that they can take up the nutrients. Another super cool microbe is Ideonella, which is a plastic-eating bacteria. It secretes an enzyme which breaks plastic down so that it can actually absorb the monomers. These bacteria have been known to break down entire water bottles over the course of a few days. Currently, we do not have a plan to dump them into the environment because that would be irresponsible. But research continues. One day you might have these be added to compost piles or to gigantic landfills to help break down the plastic, which does not break down by itself. You also have one of my favorites, Geobacter. Geobacter is a metal-eating bacteria, and while it eats that metals, the ions actually produce an electric charge. That's right, this bacteria is capable of producing electricity. There are talks about turning this bacteria into some sort of battery, which would be very, very cool. The next thing we're going to talk about is wastewater treatment, which is a type of bioremediation. We are taking our waste and using microbes to process it. When you flush the toilet, you may never have thought about what would happen. Hint, Nemo would not make it to the ocean. Instead, it goes through the waste treatment system, where first a large bar screen removes trash and large solids. This process slows down so that heavier objects can also sink to the bottom. And then you now have 2 different types of waste: solid waste and liquid waste. Liquid waste is aerated with microorganisms that will feed on the waste. These microorganisms chomp down and break down all of those waste materials into harmless monomers. They will settle to the bottom of the tank after the aeration is turned off. Disinfection with bleach will kill all of those bacteria. A dechlorination cycle will detoxify the bleach, and now the end product that is being released back into the environment is 85% cleaner. These solids are removed, and they are also treated with bacteria, but instead of aerobic treatment, this is an anaerobic treatment. So the anaerobic bacteria are put into the organic sludge, and the material is spun and spun and spun. The end product is a nutrient-rich biosolid that can be used as fertilizer. This process is incredibly clean, taking all of our human waste and turning it into fertilizers, which can be used to grow foods, and clean water. We also use bacteria to monitor water. So all organisms that poop have bacteria in their poop. All warm-blooded mammals have fecal coliforms, including E. coli, in their poop. Standard tests are to look for coliforms. Coliforms are feces bacteria. Well, coliforms are, and then fecal coliforms include 6 species that are found in waste and sewage. E. coli is one of them. EPA standards, drinking waters can have 1 coliform per 100 milliliters and 0 E. coli. Swimming water can have 200 coliforms per 100 milliliters, and I believe it's 6 E. coli, and that's it. Anything higher than that, and it is considered unsafe. You may not know, but we use microbes to make our food as well. That process called fermentation, which a lot of facultative anaerobes are very good at, produces yummy side effects. Yeast, mold, lactic acid, and acetic acid bacteria are the microbes most commonly used in food production. We classify these based on what the output is of fermentation. So you have yeast, which is used to make CO2 and ethanol. Saccharomyces is the most common type of yeast used, and it is typically found in wine, beers, and cheeses. You have molds, which are often found in cheeses as well. These will produce some lactic acid, and then your lactic acid bacteria produce lactic acid, and your acetic acid bacteria produce acetic acid. So if you want to have vinegar, you would want an acetic acid bacteria. If you would like to produce some nice cheeses, you would want a mold. So those blue and green colors in cheese, those are actually mold. One of the things we worry about as microbiologists is food spoilage. So you don't want to have harmful bacteria. While we're using bacteria and other microbes to make our food, we don't want harmful bacteria in our food. This is a really bad thing. Typically, what happens with food spoilage is overgrowth. Very few foods are sterile, so they all have some kind of bacteria or microbe in them, and then this microbe overgrows to the point where it is now harmful for your consumption. Conditions that control overgrowth are temperature, pH, water, and oxygen, and it depends what type of food you're talking about as to the correct way to treat it. So this can be very confusing. How do I know how to keep my food safe? Thankfully, the FDA has us covered. So the USDA and Food Safety Inspection Service and the FDA have come up with this app called Food Keeper. You can actually download this app and it will tell you exactly how to store your food. Basically, there are 4 steps. Keep it clean, keep your area clean, avoid cross-contaminations by separating your food. For example, you don't want to be cutting fruit on a platter you just cut raw chicken. Cook your food to the appropriate temperature. After cooking, keep things chilled to prevent overgrowth. Usually, when outbreaks happen, they tend to be related to cross-contamination or to improper storage after cooking. So let's say you make a beautiful pudding, it's amazing, and then you leave it out at the family picnic all day in the sun. You are going to be growing those bacteria, and family that eat it later in the day can become violently ill. So it's important to properly store your stuff by chilling afterwards. Foodborne illnesses are one of the top things that the CDC used to cover until coronavirus, that is. But we've had several different programs signed by different government officials to ensure that foodborne illnesses outbreaks can be contained. There's more testing that's done. They tend to track it better, and they're more easily able to find the locations. But the top pathogen food pair outbreaks are typically fish, poultry, undercooked dairy, and shellfish. So if you want to avoid those items, make sure you are cooking and storing them properly. Additionally, it might surprise you to know that salmonella can outbreak in things like peanut butter. So if a plant gets contaminated and this is not caught early enough, products can go forth and it can be a real problem. So that's why we have so much oversight on our food, is to keep the population safe. agricultural microbiology is not something people typically think of when they think of food. We tend to think of microbes that give us good food, right? Make food such as cheese or beer or bread. But we don't think about the fact that all organisms have microbiomes, including our crop species. So plant microbe symbiosis is incredibly important for healthy agricultural practices. You have fungi, rhizobacteria, nitrogen-fixing rhizomes, which are naturally part of the plant's life cycle and are required. You can have natural bioremediation and nutrient recycling by the soil bacteria. And a lot of companies have started to understand that it's not just about throwing seeds in the ground, but making sure that the proper soil microbacteria is present to allow those symbioses to occur and to prevent some pest pathogens. There's actually some microbes that reduce drought stress by providing moisture to the plant. There are some that help it with heat stress that are good at recycling, so can cut down on nutrient limitation. And to that end, one of the companies has actually started making, this is Bayer, has made crops where the seed is actually coated in a bacterial spores so that you plant a healthy microbiome when you plant your seeds. And this is a really cool, interesting way to look at increasing crop yield by using microbiomes to cut down on things that tend to impact our yield negatively and provide it with the best, healthiest soil microbiome possible. So this is up and coming. This is really, really cool stuff, in my opinion. Well, that brings us to the end of our applied microbiology sector. Exam 3 is coming up. It's April 22nd. It will be open 8 to 5. You only get 50 minutes. Disease presentations will begin on April 24th. Quiz 8 is going to be opening soon, and it will be closing on May 8th. So I hope to see everybody under the sea later on.